produce in this kind of cross? Is 9 to 3 to 3 to 1. Sure that. So, okay, so this is when we are working with two loci. Now, what happened, you know, here we have the same situation. We are going to have an organism that the phenotype that is black and short hair is not telling us too much about the genotype, because again, we have here this allele that is dominant and is masking brown. Here we have this allele that is dominant and is masking this, that is the short hair. So the long hair. The short hair is masking long hair. So what we do to find out about the genotype, we are going to do a cross with a homozygous recessive. This is going to be a double homozygous recessive for the two loci. What is the name of this cross? In this case, because we are working with two different loci, it's a two-pointed cross, right? Okay, so how many different types of gametes this individual can produce? 4, because we are here in the same situation. And what about this one? What about this one? Okay. Good. So then we have here. So what are, you know, which kind of results we expect from this cross? Because we have 1, 4, 1, 4, 1, 4, 1, 4. If we put numbers, you know, let's say that we are counting the colors of the Guinea pigs and we are measuring if they are long and short hair, we are going to expect that every single phenotypes are going to be represented in the same number. So we are going to have that the black short hair, we are going to have the same number as this black long hair, and so on. Okay? So here we are working with two different loci. So what happens if when you go to count, you know, the number of black and brown Guinea pigs and short hair, long hair, you have something that is different to 1 to 1 to 1, 1. What is going to happen? Link genes is going to mean that these two different genes that we are working with, and the genes are going to be the gene for the color of the coat, you know, the alleles are going to be black and brown. The other gene is going to be related to the length of the hair. The alleles are going to be short hair and long hair. So if here we found something that is different to, you know, the same proportion in numbers or in proportions, here it will be 1 to 1 to 1 to 1. If we find something that is different to this situation, is that we have linked genes. Can we talk about linked genes when we are working with the monohybrid cross? No, why? Because if it's only one, that's it. Now, here we have two, they can be linked or unlinked. How we recognize if they are linked? Well, here, first, if we have here the same number or the same proportion, 1, 4 to 1, 4 to 1, 4 to 1, 4, or 1 to 1 to 1 to 1, this is going to mean independent assortment. So this is going to be the second law of Mendel, right? So what it means, independent assortment, how do you explain the phenomenon of independent assortment? What it means? Do you have any mechanism to explain independent assortment? Yes, the loci are going to be distributed at random, independent one from the other, during which process? During meiosis, during the production of the gametes of the organism. So when we have two different loci, because we are working, here we are going to be working with two different genes, we are going to have that it can be an independent assortment. So if we have an independent assortment, we are going to expect the same numbers here. So what happens if this is what we observe? You know, we have all the cross there, all the guinea pigs. So when we count, we found that we have here these that are the black short hair. We have more of these than what we have of the black long hair. Oh, and what happened here? Look, here you have the brown and short hair that also you have little, and here again, the ones that are homozygous recessive, the brown long hair, you have a lot. What it means? Yes, it means that these two, loci that we are working with, they are close together. Too close together is one next to the other? What do you think? How close they are? Oh, how do you know that they are not, say, one next to the other? So this is the situation where we know that they are linked, but they are not one next to the other. They are not completely linked, because if they were completely linked, so then this is the situation that you have when you have linked genes. So if you have the situation where you have complete linkage, we are going to have that we are only have these two categories of phenotypes. So, and why is that? Because when we talk about the gametes, we are going to characterize these gametes as two different ones.